you have a heavy book or say a heavy glass of water or jug of water, and you're going to set it down slowly, and your muscles are actually lengthening despite the contraction, despite the fact that you are contracting the muscles, this allows you to lower a weight gently. It allows you to set down a heavy object without dropping it. The series elastic component. You have non-contractile parts of your muscles, your tendons. They must be excuse me, they're pulled tight when muscles relax. They're elastic, they resist distension, and they snap back to resting length. Tendons absorb some of the tensions as muscles contract, so they give you some springiness. Muscle strength is determined by the number of muscle fibers recruited, the frequency of the stimulation, the thickness of each muscle fiber, and the initial length. Now, the initial length of a muscle fiber at rest is when the muscle is at its strongest. Tension is maximal when the sarcomeres are at the normal resting length. Increasing the sarcomere length actually many times will weaken the muscle, decrease the tension. If you've got your arms locked way out and you're trying to lift where your sarcomeres are stretched, as opposed to having your elbows slightly cocked where your sarcomeres aren't stretched, you have more muscle power. Your ability to lift is more if your muscles are cocked a little, your arms are cocked a little bit, than if it's stretched and the sarcomeres are stretched beyond their normal length. Decreasing the sarcomere, decreasing the muscle length, decreases the tension and allows the muscle to go back to a normal or maximum strength. And there's a shot of the muscle tension relationship. Okay, here we are at a composition of blood, and we may skip ahead a little bit here. Let's just go through this. In the composition of blood, you're talking about chapter 14 in the lab. You've got your blood sample, you have your blood smears. Here you can see the different types of blood cells: red cell, white cell, platelet. And hopefully this is coming through on yours. Plasma, that's the fluid part of the blood. As we talked about in class, the difference between plasma and serum is that serum has been allowed to clot, which you're losing the fibrinogen and some of the clotting factors in the blood. Plasma contains albumin, as we talked about when in lab 2 or 3, osmosis, movement across a cellular membrane, diffusion. Albumin keeps drawing fluid out of the tissue, back into the lymphatic system, back into the bloodstream, goes up, it's recirculated, it's recycled, it goes through the kidney, it's filtered, and reabsorbed, cleansed, waste products are gotten rid of, and then this water is returned. But the albumin in the plasma draws this water out of the tissue. If you have somebody with something like a nephrotic syndrome where they're losing all their albumin through the kidneys, they get bloated. They look like the Michelin Man. And at that point, we would pump albumin into them, and we would pump a lot of albumin into them along with some steroids, and try to get this disease under control. Gamma globulins are antibodies. They contain they're antibodies. They help in fighting infection. You'll get more into that in chapter 15. Composition of the blood. You have the erythrocytes. They carry oxygen. They lack nuclei and mitochondria. You have a biconcave disc. Remember, we saw those. Have a 120-day lifespan. They contain hemoglobin and transferrin. Now, also, red cells do not use glucose. They carry it around to the body. They use other alternative forms of energy. and the glucose they carry to the body, or the glucose that they do not use glucose because it is in the plasma around them and carried to the body. Here is a description of formed elements in the blood. That's in your book. Okay, now we're structure of the heart. We're looking at chapter 12, lab chapter 12. And you have the atriums. The right atrium receives deoxygenated blood. The left atrium receives oxygenated blood. Right ventricle pumps a deoxygenated blood to the lungs. You know, the only artery that carries deoxygenated blood. And then it comes back through the pulmonary vein, which is the only vein that carries oxygenated blood. Like I told you, that's always a good test question. Structure of the heart. You've got a fibrous skeleton. Your heart is a big muscle. It's got a fibrous skeleton. It separates the atriums from the ventricles. The atria therefore work as one unit. The ventricles work as a separate unit. That forms the annuli fibrosi, which holds the heart valve. You've got, kind of the sockets that hold the valves in there. Pulmonary and systemic circulations. You have two different circulatory systems. You have the pulmonary between the heart and the lungs via the pulmonary arteries and back through the pulmonary veins. Remember, a vein goes to the heart, an artery goes away from the heart. You have the systemic. The systemic is between the heart and the body tissues. Blood pumps to the body via the aorta. It returns to the heart via the superior and inferior vena cava. Here is a diagram of the pulmonary system, and that is also in your book. Heart valves. You have heart valves that control the flow of blood through the heart, keep it from backflowing, make sure it's being pushed out into the arteries. You have the tricuspid between the right atrium and the ventricle. You have the bicuspid between the left atrium and ventricle. The semilunar valves are located between the ventricles and the arteries leaving the heart. Pulmonary between the right ventricle and pulmonary trunk, and the aortic between the left ventricle and aorta. And here is a diagram of the valves. Okay. Your heart is, when you listen to the heart, lub dub, lub dub, lub dub. The lub is a closing of the atrial ventricular valves. That is, as we get in the blood flow through the heart, that's the blood flows in from the veins into the atrium, the left atrium or right atrium. The right atrium is taking blood from the body. It flows from the atrium into the ventricle, the right ventricle, where it's pushed out into the pulmonary artery. The pulmonary artery carries this deoxygenated blood to the lungs, where it's oxygenated, comes back via the pulmonary vein, dumps into the left atrium. The left atrium, an atrium, if you think of an atrium in a church or an atrium in a building, it's the entrance way. And the blood flows in there, and it builds up in there. When it builds up enough, then the valves open and it flows into the ventricle. And about 80% of the blood will just flow by gravity from the atriums into the ventricle. But the lub is the closing of the atrioventricular valves. The dub is then the closing of the semilunar valves after the ventricles have contracted and push the blood out into the aortas. A heart murmur. Abnormal heart sounds are produced by abnormal blood flow through the heart, and generally it's because of defective heart valves. Sometimes you'll have a hole between, or a fistula between the atriums or the atrium and the ventricle. Mitral stenosis, the mitral valve calcifies and impairs flow between the left atrium and ventricle, and you may get pulmonary hypertension at that point, where because the flow is impeded, tension builds up in the pulmonary vein, and and then starts to back everything up, and this causes problems. A few years ago they had the diet drug fen-phen, and one of the side effects of this, in at least enough patients to get it recalled from the market, was it increased pulmonary hypertension. Another one way that they do treat pulmonary hypertension in male or female these days is to give them Viagra, which causes the vessels to relax and expand. In heart murmur again, incompetent valves that do not close properly. Instead of hearing the lub, you'll hear the lub, and then you'll hear the backflow. Or you might have a septal defect, a hole between the intraventricular or intraatrial septum, and the blood is simply flowing through that hole, and you can hear that. You can hear that with a stethoscope, and that again is another type of heart murmur. Here is a diagram. As you see, the blood is not flowing as it should be. It's kind of gurgling and back flowing. Okay, cardiac cycle. Cardiac cycle is a repeating contraction of and relaxation of the heart. Systole is the contraction of the heart muscles. Diastole is the relaxation. So when you're reading your blood pressure in 120/80, systole is 120, 80 is the diastole, where it's, systole is 120, yeah, and the 80 is the diastole, where the heart muscles are relaxing. And here is a diagram of the contractions. Cardiac cycles. The ventricles begin to contract, the pressure rises, and the AV valves close. The valves, the semilunar, the tricuspid, they're just kind of a flap, and as the pressure on the backside increases, it pushes the flap back and encloses the valve. When the pressure is low, then the valves open and blood flows in. Anyway, in the cardiac cycle, ventricles begin contraction, the pressure rises, and it pushes the AV valves closed. The pressure builds, the semilunar valves open, and the blood's ejected into the arteries. At that point, the pressure in the ventricles falls, and as we talked about even in chest expansion and that, nature abhors a vacuum. So as the ventricles relax after we push the blood out, the pressure falls, it creates kind of a vacuum, it causes the semilunar valves to close, they close, and then the AV valves open, and blood flows from the atrium into the ventricle. The pressure in the ventricles fall below that of the atrium, the AV valves open, the ventricles fill. The atria contract just at the last second, sending the last bit of blood. About 80% of the blood that is in the atrium will go simply by gravity into the ventricle. And the atria contract, sending that last about 20% of the blood into the ventricle. And at that point, the pressure in the ventricle builds up enough that it closes the valves and then opens the semilunar valves as the ventricles contract, and the blood goes out to the body through the aorta. And here is just a diagram of the cardiac cycle and pressure. Okay, the electrical activity of the heart and the electrocardiogram. Again, what we're looking at here is the tissues around the heart all the way out to the skin contain enough moisture and electrolytes that the electrical activity of the heart is projected to the skin, and it can be read on an EKG machine. And what we're doing with an EKG is essentially painting an electrical picture of the heart and the flow of current through the heart. Cardiac muscles are interconnected by gap junctions called intercalculated discs. If you look at a photomicrograph of cardiac muscle, you can see the intercalculated discs. This allows the current to flow through the heart without flowing necessarily through neurons. It's a neuromuscular type flow, and